talking. Thank you very much for inviting me, Vitaly, to give a talk. And I feel so old. Was it really 22 years ago? Oh my God. Anyway, so I'm going to tell you a little bit about the work we do. It's very applied and focused on pharmaceutical problems. So it's a little bit different from what you've been hearing this morning. And I really enjoyed listening to the last talk. So we really are in our lab, we're all focused on trying to control the transport of drugs in vivo. So we want to be able to control where the drugs go, because in that way we can make the drug molecules more effective, the medicines more effective, and then give the patient a more effective therapeutic result. So that's basically what we do in our laboratory. And our tool of choice, of course, is nanotechnology. So I'm just trying to move the slides on. Okay. So first thing to do is to thank everyone who I've ever worked with. These are all the names that I could remember. So if I've missed someone out, please don't hold it against me. I've collaborated and worked with so many excellent scientists and over the years. And the work has been funded by the EPSRC. First grant actually was 1998, and I've had continuous funding over a quarter of a century from the EPSRC. There are other funders like the Wellcome and the Royal Society and various commercial organizations as well. So moving on, and my main collaborator is Andreas Schatzlein, who's also at UCL. I'm at UCL as well as being a Chief Scientific Officer for the spin-out company that I co-founded with Andreas called Nanomerics. So, and I'll be moving to Cambridge in the autumn to become president of Wolfson College, Cambridge. So I'll be leaving UCL fairly soon. So these are all the people that I've worked with. Really, really grateful to have been able to work with such excellent scientists. So this is the talk. I'm going to talk to you about the problem we're trying to solve. And I've kind of indicated a little bit about what that problem is. I'm going to talk about the tools we use. And then I'm going to give you some case studies of what we've been able to achieve with our formulations, and then go on to talk about the commercialization activities just briefly. And the most important thing to bear in mind is that we always have made our own drug delivery systems from scratch. So I remember designing the molecule that is now going into the clinic, drawing it on my notebook, because in those days we did draw with a pen and pencil onto paper, and then deciding on how to synthesize it. And, you know, we had great people like Vitaly working in the laboratory with trying to sort of get these things to fruition, and a whole bunch of other people as well, as you've seen. So what's the problem we're trying to solve? So in order to understand this problem, the first thing we have to bear in mind is how drugs are developed. And a lot of you may already know this, but I'll just go through it for those that are not familiar with the drug development process and timelines. So the first thing that happens is that a target is identified. A part of the biology that is unique to the disease is identified by biologists, and this is an incredibly difficult undertaking. But and sometimes it happens due to serendipity. Sometimes it happens due to a large amount of money being thrown at the problem, with lots of people working to identify the area of biology that is altered by disease, be it certain cell signaling pathways that are changed in oncology, or various areas of inflammation that are upregulated in a whole number of inflammatory conditions. Once that has been identified, the target, then there are a lot of molecules that are thrown at this target in order to have a difference, in order to bring the biology back to the healthy state. And what happens is that sometimes up to 5,000 molecules are thrown at the problem before just one molecule makes it into the market and into approval as an approved drug. So it's an incredibly inefficient process. But once the lead candidate has been identified, then and identified normally in cells, or its ability to inhibit certain enzymes, for example, then we have the preclinical proof of concept work, which happens in vivo in experimental animals. And sometimes that happens at the same time as the formulation development, but sometimes the preclinical proof of concept with the drug being dissolved in any solvent that we can get away with and delivered to the animals. We work in the formulation area in that we then try and see whether we can control where the drug goes in order to improve the therapeutic efficacy and reduce the incidence of side effects. Once we have a working formulation, then we do our regulatory toxicology studies in 2 species. This is mandated by the regulator, the FDA, the MHRA, or any of the other national bodies, Chinese FDA, any of these regulatory bodies. Sometimes the formulation doesn't happen before we do the regulatory tox, but this is the general sequence of events. Then there's GMP drug product manufacture, which will involve manufacturing the drug substance to GMP, good manufacturing practice standards, so that everything is controlled and there are no surprises. Everything is reproducible and easily controlled in good manufacturing practice quality standards. Once we have the drug product, which is the drug compound and formulated as the candidate medicine using formulation strategies, and that's where we're involved, we then go on and do our safety studies in humans. And the safety studies are really to make sure, before we look for efficacy in humans, we make sure that we are doing no harm. That's followed by the phase 2 efficacy studies, where we're looking for efficacy in human patients and trying to determine by how much we can reverse and modify the disease. If those numbers are good, we move to the phase 3 studies. These are pivotal studies that we use to register the drug, the medicine candidate. And in these studies, what we are trying to see is what level of response do we get and how many responders do we get. So those of you that are familiar with the COVID vaccine story, is always good to reference this, because there's lots of interest, and lots of people were getting interested in science when we're talking about COVID vaccines. You'll know that the COVID vaccines that were eventually approved had over 90% response rates. So just looking at the control group and the group that was given the vaccine, the amount of COVID disease in the vaccine group versus the control group, we are talking about a 90+% difference of efficacy and effectiveness protecting from the COVID disease. But in some cases, just 30% of patients respond and the drug is still approved. And that happens when we have nothing else for that disease. So you can think about conditions like certain cancers, where you look at the metastatic disease and you're getting 30% responders, and that's still a good result. You can look at things like some of the lacquers that are applied to the nails to control fungal infections, where you really have very low response rates. This is an expensive process. It's very slow, about 15 years in total, and over £1 billion from concept to market. And it's very risky because there are many, many points of failure. So even if you do get to the clinical stage, there's only a 10% chance of success. And there are lots of reasons why we have failures, and majority of the failures happen in phase 2 and 3. And as you can see from the little panel, I'll just try and get my pointer if I can. As you can see from this panel here, you have most of the failures down to a lack of efficacy. And a significant factor here is that we just do not get enough target tissue exposure compared to exposure in other areas. So these are the problems that we are trying to solve. We're trying to solve this problem of very poor efficacy along this timeline, and also very poor efficacy specifically in this space. There are others that are trying to look at determining which compounds are likely to be successful using machine learning algorithms to try and predict where you're going to have success. And lots of companies have raised millions of dollars on that promise. So we'll see what happens there. But what we're doing is just trying to get more of the drug to go to the target tissue. The other problem we're trying to solve is that not many people know this, but patients just don't take their medicines. They don't take their medicines at the right time, in the right dose, and sometimes they don't take their medicines at all. And an estimated 50% of patients on chronic medication just don't take their medicines appropriately. And a high level of mortality is attributed to poor patient adherence to the medicine regime. And one thing that is clear is that patients are worried about side effects, adverse effects, and this impacts on their adherence to the medications. So this is another problem we're trying to solve by reducing side effects. And one way which we could do this is to get more of the drug to the target tissue. So let's look at the kinds of things that we're interested in. So what we're interested in is delivering molecules to the brain. Very difficult to get molecules into the brain if they're not lipophilic and below 400 Daltons. But I'll show you data where we deliver genes to the brain and have gene expression in the brain. And genes, as you know, millions of Daltons. But we've been able to achieve this. I won't show you this data, but it's very difficult to get drugs into the bladder because the bladder is constantly voiding. And so you have low residence times in the bladder. And this is one of the reasons why bladder cancer is particularly difficult to treat. But we've been able to have gene expression in the bladder as well. We've also got technology to deliver drugs to the back of the eye, the retina, using eye drops. Today, if patients have retinal diseases that are treatable, they would have an injection into their eyeball. So you can imagine a physician or a nurse approaching you with a hypodermic needle ready to inject into your eyeball. So quite unpleasant. So we've been able to show that we can deliver drugs to the retina using eye drops. We can also deliver to specific parts of the cell, for example, the cell nucleus, and that's immediately demonstrated by delivering genes, for example, to the brain and seeing protein expression. And we use these nanoparticles for our tools. So let's start with the first case study, nucleic acid brain delivery. Like I said, it's very difficult to deliver drugs to the brain because of the blood-brain barrier. The blood-brain barrier is the barrier between the capillaries that flow through the brain and the brain parenchyma. And these capillaries have incredibly tight junctions, so drug molecules cannot just float through in between the cells. Drug molecules are not taken up by the cells also because they have very low uptake activity, what we call endocytosis or pinocytosis. Very low uptake activity. They have membranes on their enzymes on their borders, which degrade any drugs that come close. And if any drugs do go into these capillary cells, they are pumped out by biological pumps, which are energy driven. So it's very hard to get molecules to go into the brain unless they're of a particular type. Molecules like alcohol do it really, really well, as we all know. And this is a problem when you're trying to treat neurological conditions, because many, many people are living with really bad neurological conditions with poor prognosis, such as brain cancers and all the different types of neurodegeneration that we're familiar with these days as we live longer. But there are other more common conditions that are also poorly served, and poorly served principally because of the blood-brain barrier, such as migraine, chronic neuropathic pain, epilepsy, and we know that also depressive diseases and psychosis. There are very few companies working in the area of mental illness using pharmacological treatments, because it's very difficult to get drugs into the brain, but it's also very difficult to determine when the drug is actually working with reliable biomarkers. So this is the first data slide showing that we are able to deliver genes to the brain and get gene expression. And this is the molecule we use. And the molecule is very biocompatible because it doesn't have a high density of amine groups, but you need these amine groups to bind to the DNA. So it's the chitosan molecule, but it's a bit different from regular chitosan, and that it's water soluble because of this ethylene glycol unit here that I'm pointing to. It's very good at getting drugs into the brain, and we go through the nose. So it's the nose to brain delivery route. And here you see a dose response. As you increase the dose, you have more of the protein signal showing. So we know that we can deliver genes to the brain by going through the nose. And we know that when we deliver these genes to the brain by going through the nose, we largely have expression in the frontal cortex, a little bit of expression in the striatum. Of course, it goes through the olfactory bulb, but you can target the cerebral cortex using this nose to brain route. We were the first to show that really you get most of your gene expression in the cerebral cortex. And this is a cartoon showing that you are delivering just to the front of the brain. And sometimes we get criticism around the rodent olfactory bulb being larger, but there is ample evidence that you can deliver molecules in solution to the brain using the nose to brain route in humans. So insulin has been delivered, oxytocin has been delivered to the brain using the nose to brain route. And we actually have a medicine candidate that we've out-licensed in this space. I'll talk about that a little bit later. So you don't get to, in our work, using particles, so we deliver particles, nose to brain, you do not get lots of expression in these more distal areas of the brain. We have a powder delivery device, which is a device that fires powder high up into the olfactory bulb, and we have out-licensed this as well with a medicine candidate to a company called Verpax in the United States. So we've shown we can deliver nucleic acids to the brain, but can we bring about a pharmacological response? And this is what we try to test here. So I'll just go through the biology to help us, to orientate us around what we're trying to do. We know that DNA damage normally starts this proliferation, and you end up with different types of cancers once DNA is damaged. But we also know that when there is DNA damage, a particular protein called P73 is upregulated, and this can cause the cells to die by apoptosis. So we know that for a fact, and this is what protects us when we have DNA damage from immediately forming uncontrollable tumors in our bodies. But there is a counter to this, and that is a protein called Itch. And this protein called Itch actually degrades P73. So in that particular scenario, DNA damage would not lead to apoptosis, and you'd actually end up then with the proliferation. So what we have shown previously with a tumor in the flank of an animal, that if you degrade this itch using gene silencing approaches, in that case, you give a small piece of ribonucleic acid, which targets the gene for itch and causes it to degrade. We know that if we do this, we get an enhancement in P73 and we end up with apoptosis. And we've shown that that causes the tumors in the flank to reduce in size. And we thought, well, could we do this in the brain? So the first thing we did was to check that if we silence the itch, we actually do get apoptosis. And that's all this slide is showing you. And so this is our delivery system here. This is our positive control, which is a lipid called lipofectamine. And these are Western blots. And what we're showing here is this is the negative control. This is a piece of ribonucleic acid that doesn't do anything. And what we're showing is that after 3 days, we've really reduced the level of itch and we've increased the level of P73. And this is a housekeeping sheet just to show that we're looking at similar levels of protein across the piece. So increasing the level of P73, I just go back. If you increase the level of P73, you should have apoptosis. And so that's what we show here. We show that once we apply this polymer with the correct piece of siRNA, we have more apoptosis as shown by this green and more dead cells as shown by the red. This is the red and green, and the blue stains the nucleus. It happens with our positive control as well, so we know the experiment is working well, but it doesn't happen with the negative control. So what we know is that, yes, with this new delivery system, which was different from the one we used in the flank tumor, we are going to get apoptosis. So we did the experiment, we put a tumor into the animals, we then gave the animals through the nose either saline, either the ribonucleic acid against the itch gene, or we gave chemotherapy, as in gemcitabine, or we gave both gemcitabine and something that's going to downregulate the itch gene and allow P73 to flourish. And these were our results. What you can see is that this is survival, the number of animals that was still okay and didn't have to be killed because they'd lost a lot of their body weight. We don't let them go to completely die. We just look at a humane endpoint, lack of reduction in body weight. So with saline, they have very low survival. When we downregulate the itch protein, they also have a little bit longer survival, but it's not statistically different from saline. That was a surprise to us, actually, because we thought that delivering this nucleic acid through the brain would have a larger effect, but it doesn't. When we added gemcitabine, we slowed down the progression of their death here quite clearly. And it was when we had both. So when we had gemcitabine damaging DNA with the P73 upregulated, and we showed that the P73 was upregulated at the tumor site, then we really have a longer survival of the animals. And here, this just shows their body weight, which is used to inform this data. And this is currently under review. So quite clearly, once you have gemcitabine and the anti-itch therapy, you have a reduction in the tumor. The tumor's around about here. If you have saline, look at the size of the tumor and it pokes through the brain parenchyma. If you just kill the itch protein with the ribonucleic acid against itch, you have a small reduction in the tumor poking through. When you have gemcitabine, you can just see the shadow of the tumor. But when you have both of them, both gemcitabine and the downregulation of itch, upregulation of P73, you have the best result. So what we've shown is that we can deliver nucleic acids to the brain and achieve therapeutic outcome. And this therapeutic outcome really is just measured by the survival days. So we extend survival by 73%, and that's very, very good compared to the control animals. So the next example I'm going to give you, and just to bear in mind that in this last example, the nucleic acid was always delivered using this polymer, this chitosan derivative that I showed you earlier. With the oncology platform, we have a slightly different polymer that we're using. We're using a biopolymer. And here's the story. Triple negative breast cancer is a very serious disease with a very poor survival. Patients diagnosed with breast cancer have very good 5-year survival normally. So a lot of us will know someone who's had breast cancer and is leading a normal life. Breast, colon, testicular cancers, these are all cancers that I would say we've largely conquered to a great extent. I mean, they're not curable, but people do have good prognosis. But with triple negative breast cancer, the 5-year survival is a lot lower. And if it's metastatic, then it's palliative care these patients are offered. So this is a very difficult to treat cancer. But we know that this particular cancer is associated with an upregulation of a particular type of receptor called the CD44 receptor. And so the thinking is that if you can develop a delivery system that targets this receptor, you might be able to improve these numbers. So what we did is that we took human serum albumin and we derivatized it. And this is, of course, under review as well. And we derivatized it, and then we encapsulated a known drug, and in this case, it's paclitaxel, within this platform. And then we sought to test it, really. And we sought to test it against CD44. And quite clearly, what you can see from this particular slide is that you have very good uptake, and this measures uptake into the cells with this particular delivery system. And the delivery system is decorated with a fluorescent tag so you can follow it. But if you add the antibody against CD44, uptake reduces, and it reduces further if you add more antibody. So really clearly what we've shown here is that we are having uptake through the CD44 receptor. If you don't have the CD44 receptor, you don't have the level of uptake. And you can look here and look at the y-axis. This is 1/8 of this actual y-axis. So we're really not getting uptake. And these are delivery systems which haven't been derivatized in the same way that this delivery system has been derivatized. So what we established, and we did this with hyaluronic acid, also as a blocker for CD44, and had the same result. And we know that these particular cells that have the CD44 receptor are also, this is synonymous with the cancer stem cells. Cancer stem cells are the more resistant cells that you can't really get rid of. So, and we demonstrated that we have better uptake into these cancer stem cells. It's not on this slide, but we demonstrated that. And this just shows you, this is a visual showing that if you have the derivatization, you really have uptake into the cells that have the CD44 receptor versus if you don't have the derivatization. And coumarin was a fluorescent marker that was encapsulated in these nanoparticles. So these are nanoparticles made from a derivatized form of human serum albumin. And then we tested them. We tested them against lots of cells, but I'm just showing you this data where we tested them in a tumor model. And quite clearly what you can see, this is the standard of care that patients will receive today, Abraxane. The tumors stop for a while and then start to grow after you stop dosing. If you don't treat, of course, they go unchecked. But with our system, what you can see is that you have a reduction in tumor growth, and the tumors then don't recur for 90 days. 90 days is about a quarter of a laboratory mouse's life. So what we are saying here is that we appear to have got the cancer stem cells with this particular biopolymer. Moving on to commercialization now. So I've given you 2 examples of how we're trying to solve the problem. And what we've done is we've spun out nanomerics, and we have what we call the molecular envelope technology. This technology won first prize in the Royal Society of Chemistry's Emerging Technologies competition in 2017. But this year we were really pleased to win the King's Award for Enterprise in the area of innovation for our company. And the King's Award for Enterprise is the most prestigious business prize you can actually get in the UK. These are the people behind the company. There's myself and Andreas Schatzlein, our co-founders. We have Andrea Maica, business strategist and a small investor. We have Alan Bayer that's taken 40 compounds from concept to market. We're trying to take one polymer from concept to market, and Alan Bayer's taken 40. So we're very happy to have him in our team. We have what we call an active excipient. We take this excipient and we use it to formulate our molecules, and then we can control where the molecules go. It's very different from the normal thing that happens. People take off-the-shelf excipients, mix them in different proportions, and then see where they go. We work in the area of nose to brain, like I've just mentioned. We also work in ophthalmology, and we have what we describe as the world's only non-irritant penetration enhancer. So we really do have penetration enhancement into the ocular tissues. And we do the intravenous to oral switch as well. So the first molecule we looked at was enkephalin, which is shown here. And we wanted to see whether we can tackle the problem of pain. And as you know, pain, 1 in 5 people are nursing a chronic painful condition. It's worse as you get older. And very few have pain relief. And most people are working with the mu opioid receptor agonists like morphine and fentanyl. And we know that they have awful side effects. You've got the constipation side effect, which happens almost universally. You have respiratory depression. You have overdoses, nausea. And of course, you have habit formation addiction. And about 110,000 people die every year in the United States from after that's connected to the use of opioids, and normally I'm talking about prescription opioids. We don't have the opioid crisis to the same extent in the UK. It doesn't mean we won't have it. Anyway, so what we thought is let's use leucine enkephalin make it druggable, so we can have a differentiated product with fewer side effects. And that was our goal. But first of all, we have to get it into the brain. And we tried with, first of all, and I noticed in the last talk, there were some lipophilic peptides. So we've made lipophilic peptides, and we've been able to make these nanofibers with these lipophilic peptides, picture here, and these sort of how we think they assemble here with the peptide bit in purple and the lipophilic part in blue. And what we found is that when we had these nanofibers and injected them intravenously, we did get the nanofiber signal in the brain. With the peptide, we could not detect the peptide in the blood or in the brain. We could only detect the metabolite, because this particular peptide loses its arginine almost immediately the moment it gets into the blood. We also found that this peptide derivatives we've made was able to give our therapeutic effect, and we test that to look at pain. We look at pain relief, we inject it, we subject the animal to a thermal stimulus, 55 degrees centigrade, and look at how long it takes to remove its tail. And then we measure that, and you can see it's only when you have the nanofibers that you actually have proper pain relief. With the drug on its own, you don't get anything. So that's fine. A good way to get the stuff into the brain is to make a lipophilic derivative, cause a self-assembly, and then administer. But the problem with that is that we don't want everybody to have to give an intravenous injection. So we needed something else. And so we instead just made nanoparticles and encapsulated our peptide. And then we made those nanoparticles into microparticles so that we could give them through the nose to brain route. If you deliver nanoparticles through the nose to brain, they would just come out in the breath stream. You need them to settle at the top of the nose. So we make them into microparticles, and to make them into microparticles, it really is like a bunch of grapes all stuck together, the nanoparticles, to make them microparticles. We found that we got the drug into the brain. We found that we had pain relief. In this case, we have an inflamed paw, and we're poking it with different pressures, looking at when the animal removes its paw. It takes longer to remove its paw once it's had the drug compared to the drug, once it's had the formulated drug in red compared to the drug in solution. And again, we're going to the nose to brain. What we also discovered is that this enkephalin formulation that we'd discovered and designed was actually didn't have any tolerance. And this is shown in this particular data set. You give morphine on the first day, and if you give morphine twice a day, by the 5th day, you're having a very small response. And 80% of patients will develop tolerance to morphine. And yet, when you have our own formulation and you give it again twice daily, you actually still have pain relief. And so we were the first to show that there was no tolerance developing. And also that this formulation works when there's morphine tolerance. So if you gave these particular animals the enkephalin formulation that we developed, you would also find that they would respond. That data is not on this slide, but that's what we found as well. So we showed that it was active in all pain models, no reward-seeking behavior, no tolerance. It was active in morphine tolerance, and it was centrally acting, so unlikely to cause constipation. And so bearing this in mind, we out-licensed it to Verpax Pharmaceuticals, and they are responsible for the clinical development. And when you compare that to fentanyl and morphine, you can see the real examples: unlikely to give constipation, unlikely to be cause respiratory depression, because it has some, as well as mu opioid receptor agonism, it also has some delta opioid receptor agonism. We showed that there was no analgesic tolerance, showed there was no, it was active in morphine tolerance, and we showed there was an absence of reward-seeking behavior in a set of experiments where we dosed the animals and we checked to see whether they went into a particular dosing chamber when they didn't have a painful stimulus, and we found they didn't. So that's been out licensed and been developed and should go into the clinic early next year. The other formulation we're looking at is for allergic eye disease, and we're doing our own clinical trial in that one. And what we've shown here is that you get a lot more drug going into the tissues compared to the commercial formulation, and you get no drug in the blood. That's a really unique capability. And that's important because you do not want systemic side effects, which you could get with the commercial formulation, where you get some drug in the blood. So this is looking at the kind of allergic conjunctivitis that's not controlled by antihistamines, so a type 4 hypersensitivity response. And we went to the MHRA and we got a fast track passport, and we're doing the phase 1 project at the moment. So we don't expect ocular irritation from our own formulation, even the suspension has a level of ocular irritation. This is just the clinical trials plan. We'll have altogether 10 subjects with given either the placebo, that's the molecular envelope technology on its own, or given the medicine. We're working with PACE to manufacture to GMP, and we're working with Quotient to do the clinical trial. And we also have a way to deliver drugs to the back of the eye, the choroid retina. You can see this in the rabbit. We always use the rabbit model because the anatomy is similar to humans. When people use mouse or rat models, it's very easy to deliver to the back of the eye in those models, but the rabbit model is the model of choice. And then here to show that the drug is actually efficacious in a form of the disease in a mouse model. And what we're showing here is that it's as efficacious as a steroid formulation. And you can't give steroids for long, because if you do that, patients are at risk of developing glaucoma or developing cataracts. So this will be a steroid-sparing formulation. And what we've shown is that it upregulates Tregs and downregulates certain other T cells. We know how it works. We know that the particles are positively charged. They're attracted to the negatively charged surface of the cells and mucosa. They're also they penetrate the mucosa and sort of adhere quite strongly to the mucosa. Once that happens, the particles are destabilized and you get the drug then going into the tissues. And for the back of the eye, once the particles get into the drug gets into the tissues and into the choroid, that's how it gets to the retina. So it's the fact that we can put so much drug into these tissues. That allows us to get to the back of the eye. So, like I said, the most important thing is that it's a penetration enhancer, which is not irritant. If you wanted to cause penetration enhancement into tissues, alcohol will do the trick. Mixing drugs with vodka will do the trick. And please do not try this, but it will do the trick, but it'll be incredibly painful and damaging to the cells. Even some surfactants will do the trick, but again, they are quite painful, and if they are going to cause penetration enhancement, highly likely to damage the cells. And you feel it if you have a little bit of soap in your eye, for example. And this is our pipeline. We have a number of medicine candidates that are making their way through the different stages. We have some that we've out-licensed and some that we're working with, some in stealth mode with some larger players who don't want anyone to know that they're working with us yet. So I hope I've convinced you that you can control where drugs go. You can repurpose, reformulate, and redirect by showing you that we can deliver nucleic acids to the brain, treat various neurological disorders, shown you a oncology platform targeting cancer stem cells, shown that we can deliver metabolically labile peptides. So enkephalin, as you know, is a peptide to the brain. We can target drugs to the ocular surface tissues with no plasma exposure, and we can deliver drugs to the retina using eye drops. And this is our manufacturing facility. We're based in just outside Wembley Stadium, North London. And this was one of our early team photos just after COVID, and we all went out for our first meal. And with that, I will end and look forward to answering any questions. Thank you very much.
Thank you very much, Ijeoma.